I hope you all enjoyed your lunch. The afternoon session of the second day of Bioplus-Interphex Korea will soon begin. It will consist of new generation CAR T cell therapies, global clinical session, and IQVIA sponsored session. The next session is new generation CAR T cell therapies. I would like to invite Chairman Lee Byung-gun of Council for Advanced Regenerative Medicine. It is great to be here. Welcome everyone to Bioplus-Interphex Korea 2021. I am the chair of this session for new generation CAR T cell therapies, and I am Chairman Lee Byung-gun of CARM, Council for Advanced Regenerative Chairman of Medicine. We have been working very hard to battle cancer, and for immune therapies, we are very much interested in CAR T. CAR T therapies, compared to other therapies, have very good effects, but it is quite expensive. And there's the cytokine release syndrome, a adverse effect, so it is not much in use yet. And it can only be used mostly for malignant tumors. So we are thinking of how to use it in the solid tumors rather than for the hematological tumors. Today, we are going to be hearing about new generation CAR T cell therapies from many experts. I would like to introduce CEO Lee Jong-seo of Epclon. He received his degree from Kyoto University and worked as a researcher at Harvard. And he established Epclon in 2017, and he has been engaging in biosynergy discovery as the CEO of Epclon in new therapies. And as a result of this antigen platform from Seoul National University and Pennsylvania University with other researchers, he has been developing a unique CAR T therapy program. He will be talking about the novel CAR T programs in Epclon. Please.
안녕하십니까.
Good afternoon, everyone. I am Lee Jong-seo, CEO of Epclon. I would like to thank the organizer for inviting me this year. Today, I'm going to talk about the novel CAR T programs in Epclon. To give you a brief introduction of the company, based on the antibodies we produce, the monoclonal antibody drugs and bispecific antibody drugs called Afovlab. And also, I'm going to especially focus on the Z-CAR-T, which is a CAR T cell therapy targeting the cancer cells. Of the products, there is the NEST, which is AC101. We have passed phase 1 in China, and we have started phase 2 for the cancer patients, and for CAR T, as was just mentioned by the moderator, for hematological AT101 and AT501 for ovarian cancer are being developed. Could you share the slides again, because your slides are static? Thank you. Let me fix that. Yeah, that was just the introduction. Can you see the slides now? Yes, we can see them now. Thank you. It's just an introduction, so I'll continue from the CAR T. I'm sure you've seen this slide many times. For CAR T therapy, you take the blood from the patient and it is delivered to Epsilon's GMP center, and Prodigy is used. T cell is selected and goes through activation, transduction, proliferation, and packaging into parenterals, QC, QA, and then injected into the patients in clinical study. And there could be some manufacturing failures, or in order to enhance the completeness of the production, we are going ahead with an automated process for manufacturing the CAR T cell therapeutics. I'd like to explain the CAR T program in more detail. First, AT101. On the surface of the cancer cell, there is the CAR that is expressed. This is the typical CAR that we usually see. And looking at the B cell development and of the processes, there are some specific cancers that can develop and diffuse large B cell lymphoma, or DLBCL, and acute lymphoblastic leukemia, ALL, and follicular lymphoma, FL. For the targets, CD19, CD20, or CD22 are available. And the widest ranging is CD19. So for hematological cancer, we select CD19 as the priority target. And as for the structure of the T cell expression, the outside and the inside could be separately divided. The target material, CD19 targeting antibodies, will be placed on the outside. There's the antibody and the target material that are combined within the cell in order to facilitate the material delivery, interferon gamma. These mechanisms are used to kill the cancer cells. But as for the CD19 CAR T therapeutics, they use an existing material. And so H12, 18, they only apply the same thing. And so for a long time, we have been applying the 1218 at Epsilon. And at the final stage of commercialization, this is quite useful. And this is the deciding factor in our intellectual property before commercialization. And as for effectiveness, the epitope is different. So compared to the other products, there is differentiating factor and also superiority. And so this is a very decisive factor. Ecronic CAR T, in the cell models, there's the mechanism and cancer cell killing mechanism that are similar. For example, cytokines are released and cancer cells are killed. And so the series of events are identical. And so there is a great degree of anti-carcinogenic effect. And an antibody is developed under special circumstances. We have tried to come up with the antibody through many routes. Chicken immunity library used to come up with the antibody. Through affinitive iteration, immunogenicity was released and optimization was done. And so we were able to get the ultimate antibody of 1218. And the 1218 antibody, in terms of binding power, in vitro FMC 63 is better, but as for the cell binding through fax or in vivo model, the binding capabilities, they are similar, or it is confirmed that 1218 is superior. As for the CD19 binding structure of 1218, we looked at FMC63 and epitope test, and as a result, it did not intervene with FMC63, and 1218 bound itself to a new area, and we have conducted additional testing, and what we found out was a structure of CD19 allows FMC63 to bind with the outside of the cell, but 1218 binds close to the membrane of the cell, and that is the difference. As for CAR-T activation using 1218, we looked at interferon gamma release. CD19 expressed a volume was different in 4 different cells, and we looked at the activity. CD19 expression was different, and we applied 1218 and FMC63 expression, and cytotoxicity interferon gamma release. They showed similar results and similar level of activity, and the amount of CD19 expression was relatively lower, and in those cells 1218 showed enough activity level. There was the comparison study of 1218 CAR-19 with FMC63 CAR-19, and we compared using an animal model. In large cells, lymphoma, and then NAM6 leukemia, we used the mice models. 1218 and FMC63 were applied, and of the CAR-T, we could see that the cancer cells were removed. So relatively speaking, the tumor burden was lower, and we tried under 2 scenarios with lower tumor burden and higher tumor burden. AT101 activity was checked, whether it was low tumor burden or low tumor burden. There was AT101 of 10 to the 6th power, and we could see that the cancer cells were removed. We are using 1218 and AT101 interchangeably. AT101 is for the cell line made in GMP facilities, and 1218 is the name that we adopted in the lab environment. From UPenn, there is Dr. Marco Ruella and his team deals with hematological cancers. In order to compare 1218, the team looked at, he built the hematological blood cancer model to compare with FMC63. And in a model in which FMC63 did not work, our H1218 still was effective, and this was announced time and again in many conferences, and we are still conducting the test. And so that was it for AT101. And let's now go into AT501, which is more technologically innovative. You must be aware CAR T is very strong in hematological cancers and it's a promising therapy, but it has its downsides. For example, CAR T after injection, if they're kept in the patient's body, after that, it's difficult to control the CAR T cells and they are very active. So we call it CRS, cytokine release syndrome, and some adverse effects they happen. And so acronym Z-CAR-T, a switchable CAR-T is being developed. Z-CAR-T, this may be a bit complex, but basically Z-CAR-T does not recognize the cancer cells directly. It recognizes the switch molecules. So when there are no switch molecules within our body, when the Z-CAR-T is injected, then the Z-CAR-T is not active. After the Z-CAR-T injection, depending on the patient's condition, if there is a switch material that is injected that allows the cancer cell detection, and then the Z-CAR-T is activated. And if you need higher level of activation, you need to have higher dose injection. But if it is active enough, then it could go with lower concentration. And if you need to stop the activation, we stop injecting the switch materials altogether so that we can turn on and off the CAR-T activation, and we can also adjust the degree of activation. There is the COT9, which is added. As for Z-CAR-T of the switch molecules, it recognizes the COT9, and the cancer proteins are discovered and to bind them. And so it can find the cancer cells in the body and can connect the cancer cell with the Z-CAR-T. So the existing CAR-Ts, independent CAR-T had to be developed for the cancer cells, but regardless of the target cancers, we can have the Z-CAR-T developed. So we just need to change the switch molecules. Then Z-CAR-T can recognize different types of cancers, and if needs be, if you inject two types of switch molecules, then you can have activation on different cancer types with the same one Z-CAR-T. AT501 is for HER2 ovarian cancer. So we looked at the Affibody library, and we have the COT9 bound together, and we finally chose the optimum switch molecule. To develop Z-CAR-T, we had to develop a mouse antibody, and after making that antibody, we humanized it and optimized it, and was able to develop that into an antibody material. And the cotinine antibody can bind directly with COT9 that can detect the cancer cells. And so through the development, HER2 switch material and COT9 was developed into a CAR-T. And so with the CAR-T developed, we looked at cytotoxicity and IFN release. And depending on the concentration level of switch molecules, we looked at whether the activation level was adjustable, and we checked it in vitro. And what was interesting during the development was the cotinine have two optical characteristics, and only one form could have the Z-CAR-T active. The final switch material is isoform 2. So we developed that switch, and we are conducting the development with that material. The HER2 switch and Z-CAR-T were used to check the activation level in a mouse model. HER2 positive ovarian cancer, SKOV3, was guided, and switch and Z-CAR-T used independently, and the two applied simultaneously. Switch molecule alone or CAR-T, when they were independently applied, there was no activation, and there was activation and level of activity against the cancer cell only when there was switch and the Z-CAR-T together. And we also looked at the correlation in the animal model. And in the ovarian model, after injecting Z-CAR-T, the number of Z-CAR-T cells were the same, and we handled the switch materials in different concentrations. And although the Z-CAR-T was the same, depending on the concentration of the switch, the tumor burden came out differently. And so with a higher concentration of switch materials, the cancer cells were killed more effectively. AT101, we applied for FDA IND in June, and we're preparing for clinical. And we are expecting the clinical study to take place in Asan. And AT501, we're preparing for the clinical trial, and we are targeting ovarian cancer, and we're developing other types of CAR-T. The CAR-T team at UPenn, Seoul National University, Seoul National University Hospital, Asan Hospital, and biotech companies G+, Fulgen, Ensage, Evixgen. These are our partners, and we are building a strong network for next-generational CAR-T products. I would like to thank Dr. Jung Jung-ho of Seoul National University Hospital for his leadership, Professor Song Young-san of SNH, Professor Marco Rilla of UPenn, and my thanks goes to many professors and colleagues. I would like to thank you for your attention. If there are comments or questions, please write me through this email address, and I'll try to answer your questions to the best of my ability. Thank you very much. Thank you very much. That was quite an interesting talk. And the CAR-T function was eliminated, and cotinine-based CAR-T system was developed, and I hope that you will have good results in your clinical study. And we do not have Q&A, so if you have questions to the speakers, please note the contact information on the slides. Thank you.
Results.
I'd like to introduce to you the third speaker. It is Dr. Ji-Hoon Park. He has majored in biochemistry and at Chungnam University Med School. From 2014, has worked at NHLBI and under NIH doing his postdoctoral, and he is now the CEO of Terra Immune. T-Rex are used and is trying to create a world in which there is no autoimmune diseases. It was founded in 2018. He's going to talk about his experience developing the CAR T, and his presentation is titled Gene Engineered T-Rex Cell Therapy to Cure an Anti-Drug Antibody Problem. Dr. Park.
My name is Ji-Hoon Park, the COO and co-founder of TerraImmune. Today, I want to talk about gene-engineered T-Rex cell therapy to treat an anti-drug antibody problem. Today, I will cover these topics: introduction of CAR-Treg, clinical applications of CAR-Treg, and complex manufacturing processes of a clinical-grade CAR-Treg. First of all, I want to talk about what is CAR. CAR is an abbreviation of chimeric antigen receptor, which is consisted of single chain Fb, which recognizes the antigen on the surface of target cells. Those antigens can be CD19, CD20, or any kind of proteins on the surface of target cells. And transmembrane domains, which helps this receptor to be bound on the surface of immune cells. These immune cells can be T cells, NK cells, or even Treg cells. And then signaling domain, which delivers the activation signals to those immune cells and make them do their functions. And these are types of immune cells. We know B cells, which is professional antigen presenting cells, and which can be a plasma cells to produce antibody against the antigens. And CD8 cytotoxic T cells, natural killer cells, which can kill or remove the target cells. Those target cells can be cancer cells, or sometimes can be a healthy cell. And CD4 helper T cell, and regulatory T cells. So today, I want to talk about regulatory T cell, shortly Treg cells mainly. This is a principle of CAR T cells, how we can use them in the clinic. We isolate T cells from the patient's blood or the third person's blood and make them to be specific to the target or antigen by introducing the receptors using the viral systems or electroporation. And then we can make target-specific T cells. We can call it CAR T cells, or it can be TCL T cells. And then we send them back to the patient bloodstream, and these CAR T cells can recognize and bind to the cancer cells or target cells. And finally, these CAR T cells removes or kills the target cells, the cancer cells. This slide shows the applications of CAR in the clinic. As I explained in the previous slide, we can use CAR T cells for the oncology. As we have seen in the great success in the oncology, CD19 and CD20 could successfully remove the cancers from the patient body. And some scientists are approaching to the solid cancers using neoantigens. And we can also use the CAR T cells for the infectious diseases by the HIV, EBV, or CMV, or and so on. We can successfully and we can selectively remove those infected cells using our CAR T cells. And allogeneic transplantations like GVHD and organ transplantation. Some group also show that the CAR Treg or polyclonal Tregs successfully inhibit the organ rejection, which happens in the GVHD and organ transplantations. And we can also use the CAR-Treg or CAR technologies in the autoimmunity or autoimmune diseases like the type 1 diabetes, Lou Gehrig diseases, or lupus and many other autoimmune diseases. So how does Theraimi use CAR in the clinic? The answer is regulatory T cell, Treg. Regulatory T cell or Tregs are specialized subpopulation of T cells that act to suppress immune responses. So they can inhibit T cell proliferation and cytokine production and play a critical role in preventing autoimmunity. And this is how we manufacture or how we prepare the CAR Treg cells in the laboratory or GMP facility. We collect the patient's immune cells from the bloodstream and isolate Treg cells using the cell surface markers and using the ClinIMAX system. Then we introduce or we transduce the target-specific receptors like TCR, T-cell receptor, or CAR using the retrovirus or lentivirus system. Then we expand them using our proprietary technologies, Treggable and Tregging, for 14 days, and we can get 50 to 100 times of CAR Tregs in the laboratory. Then we infuse those TCR or CAR Tregs into the patient's bloodstream to treat the diseases. Using these technologies, we are developing 6 pipelines in total. The first pipeline, TI-16A, is targeting hemophilia A using factor VIII TCR Treg, and TI-235 for multiple sclerosis. And B-cell lymphoma is in our pipeline, xenotransplantation, atopic dermatitis, and rheumatoid arthritis is under development. And our primary pipeline, factor VIII TCR Treg for hemophilia A, is in the preclinical stages, and we are planning to submit the IND package in the United States in December this year. And we will recruit the first patient in second quarter of 2022, next year. And I will explain a little bit more about our primary pipeline. Hemophilia A is a genetic disorder, and which it has the patients has the factor VIII mutation in their genome, and they cannot produce factor VIII proteins. So the first line treatment for the hemophilia A is a factor VIII replacement therapy. It means the delivery or injection of recombinant or purified factor VIII protein onto the patients. Most of the patients can be treated with that factor VIII replacement, but some of them develop anti-factor VIII antibody, which neutralizes the effect of the therapeutic factor VIII. So the only way to make them healthy is to increase the factor VIII doses or inject factor VIII proteins more frequently. It means there is no cure for factor VIII antibody complications. This is mechanism how our CAR Tregs can treat the anti-factor VIII antibody problem. The antigen-presenting cells in our body processes the factor VIII protein, and T-cell B-cell cascade induces the production of anti-factor VIII antibody. Our Treg cells with TCR or target-specific receptors can move near the immune cascade and shut down the cytokine releases and signaling cascade, and finally shut down the anti-factor VIII antibody production in the patient's body. We could show the in vivo efficacy successfully and which was published in Blood Journal in 2017. As you can see here, once the hemophilic mice are exposed to the factor protein, they immediately develop the anti-factor antibody from their body. But once those mouse were injected with factor VIII TCR Treg or factor VIII CAR Treg, we can see the anti-factor VIII antibody production was attenuated or prevented by our Treg cells. This is our development milestones. Since 2018, we were awarded the SBR Phase 1 grant from NIH, and we were also awarded PACT program from NIH, which supports manufacturing processes and regulatory affairs for the IND submission. In 2021, we could set up the manufacturing processes in the GMP facility, and we also set up an executive advisory committee for clinical trial. And we are planning to submit the IND packages for phase 1 and 2A clinical trial in the United States in December this year. And we will also complete the clinical protocols in the last quarter of this year. Another part Theraimune is focusing on is to establish standard manufacturing processes for CAR-Treg. As you can see in this slide, the manufacturing processes are very complex and requires hands-on experiences. The manufacturing processes has more than 20 steps through day 0 to day 14, and we have 4 QC testing points, and we also have very strict acceptance criteria, including sterility testing and Treg phenotype analysis. These processes are very complex and know-how accumulated processes, and these are all required to manufacture safe Treg cell products in GMP facility. And we are trying to concentrate on to establish automated cell manufacturing in closed system using the Miltenyi Clininex prodigies. So with that effort, we expect less amount of labor and effort and lower chance of contamination and more reliability and reproducibility of our Treg cell products. With these efforts on the development, we believe future therapies for autoimmune diseases are Tregs. Comparing the traditional immunosuppressive reagents like small molecules, antibodies, and biologics, which are systemic immunosuppressants during the long-term treatment, can cause serious risk of infection and potential secondary tumorigenicity. However, Theraimune Treg cell therapy uses natural immune cells and targeted natural immunosuppression by inducing cell-mediated immune tolerance. And this disease targeting immunosuppression by engineered Treg cells may have no or less adverse events over the years. Thank you for hearing my talk today. And if you have any questions, and if you want to learn more about our technologies and pipelines, please contact me at jpark@theraimune.com, or please contact Mr. Kyuno Han at Theraimune Korea office. Thank you so much.
네, 박준.
Thank you, Dr. Park. Treg under development. I hope that you will be indeed able to create a world without autoimmune diseases.
재즈 회로
The fourth presenter is Jazz Harrop and is currently a VP of product licensing at Oxford Biomedica. And to my knowledge, Oxford Biomedica is a spin-off of Oxford University. And in Kimria, there is the antivirus that is supplied by Oxford Biomedica. Jazz Harrop, before joining Oxford Biomedica, worked at GSK as a senior director at platforms an interim head of rare diseases and also was senior director at Biopharmaceuticals Business Development. He will be talking about the limitations of the current CAR-T treatment and how to overcome solid tumor treatment, and challenges and opportunities for CAR-T therapy is his theme. Please.
Hello, it's a pleasure to give a presentation at the conference on the topic of challenges and opportunities for CAR-T therapies. My name is Jez Harrop. I'm a VP of Product Licensing in the Business Development Group at Oxford Biomedica. I'll give an introduction to Oxford Biomedica initially, and then go into talk about CAR-T therapies, a potentially novel and new class of products, discuss some of the recent approvals in this area, and then some of the successes and the key challenges. And lastly, I'll be providing an update on some of the novel technologies which could help to expand the uptake of CAR-T therapies in the oncology space. So Oxford Biomedica is a fast and growing cell and gene therapy company. It's a leading company in the sector, mainly focused on lentiviral vectors. OXB's vector was used in the first FDA-approved lentiviral CAR-T program, developed by Novartis. The brand name's Kymriah. This is a CD19 CAR-T targeted for hematological cancers. We have additional partnerships with other leaders in the field, including Bristol Myers Squibb, Shire Gene Therapies, AstraZeneca, and Boehringer Ingelheim. OXB's improving CDMO revenues are providing a good sound foundational basis for funding our proprietary pipeline, which is going to be a major focus going forward. OXB is in the FTSE 250. We have around 18 partner programs with our CDMO customers. We have our own proprietary pipeline and over 670 staff now located in 6 facilities in the UK and the US. So OXB obviously has a leadership position in the lentiviral field, mainly through, as I mentioned, our collaboration with Novartis on their CAR-T program, Kymriah. We are the sole commercial supplier for Kymriah globally. And this is now approved in over 38 countries. We have expanded our relationship with Novartis recently to include 5 additional CAR-T products, and we've extended our commercial supply agreement for a further 5 years. We have brought on new CDMO partners in the CAR-T field, including BMS Juno for CAR-T and TCR-T programs, and also Beam Therapeutics for a single CAR-T program, which could expand to other CAR-Ts going forward. And we are continuing to explore relationships with other leading CAR-T companies. So this is the OXB pipeline. So we took an AXO-Lenti-PD Parkinson's program into the clinic. That was then subsequently licensed to Shire Gene Therapies around 3 years ago. And our fully owned proprietary programs include OXB302, which is a 5T4 CAR-T product targeting an oncofetal antigen widely expressed on hematological and solid tumors. The lead indication we're taking that forward in is AML, or acute myeloid leukemia. And then earlier in the pipeline, we have a number of in vivo lenti liver programs, which haven't been disclosed yet, which will be also a major focus for us going forward. So switching gears to CAR-T therapies, this is a totally novel class of product that has made a big impact in the last few years. The manufacturing process is quite complex. Cells are taken from a patient's blood. They are then purified and then exposed to a lentiviral vector or a gamma retrovirus vector, which then transduces the novel chimeric antigen receptor into those T cells. These CAR-Ts are sort of synthetic antibodies which allow you to bind to specific tumor antigens. Those CAR-Ts are expressed on the surface of the transduced T cells. Those T cells are then cultured in vitro and expanded, and then reinfused back into the patient. And the CAR-T, the T cells expressing CAR-T, then go and target the specific tumor and kill the tumor cells. in many cases leading to a complete resolution of disease. So there's been a number of approvals in this space in the recent years. So we mentioned Novartis were the first to get FDA and EMEA approval, but there's been followed closely by Yescarta, Breyanzi, and Tecartus. They're all approved for hematological tumours. And then more recently, Bristol Myers Squibb has got approval for Abecma, which is a BCMA CAR-T targeting relapsed refractory multiple myeloma. So we see this field expanding in the future, but there are, I guess, some interesting data here in terms of sort of the efficacy of Kymriah in B-cell acute lymphoid leukemia. And we're seeing here overall survival of 70% at 18 months post-infusion. And actually, that's maintained and stabilized out to 24 months, seeing 66% overall survival at 2 years. So it's a very promising class of therapeutics. But there are some challenges with this type of therapy. They're very powerful therapies. There are safety issues associated with the therapies. There is a unique set of toxicities that are seen, most commonly cytokine release syndrome and neurotoxicity. And these conditions, these safety issues can be potentially fatal. Another concern for the class is just the cost of therapy and potential patient access. These currently are autologous CAR-T therapies, so they're very expensive. Currently, Kymriah and Yescarta at $475,000 and $373,000 a listed price in the US. So this is a barrier to treatment for all but the most severe or refractory patients. I think another challenge is the limited efficacy seen so far in solid tumours. To date, efficacy has mainly been demonstrated in hematological tumours. There are challenges in treating solid tumours because of their immunosuppressive microenvironment, which can induce T-cell exhaustion. We have failure of the T-cells to penetrate the tumour tissue. And autologous treatments are unlikely to deliver sufficiently larger dose of CAR-Ts to be able to treat solid tumours, particularly larger solid tumours. So there are some issues in developing the platform further into these kinds of therapies. So some of the solutions for safety are that it's now becoming standard practice to treat patients with immunomodulators, such as the IL-6 receptor antagonist tocilizumab. Also, there are kinase inhibitors that can be used to treat patients that are undergoing safety issues with CAR-Ts. And this can actually down-modulate the signaling in CAR-Ts, stopping them becoming a kind of a major safety concern. It's possible to integrate on-off or suicide switches into the CAR-T construct, which allows you to modulate, activate or suppress the CAR-T or even induce lethality in the CAR-T cell, preventing further potential safety concerns. And there are also vectors that can have stimuli-responsive CARs. These can be CARs that are masked with a peptide and a protease cleaver, and they're only activated in the presence of enzymes that are secreted by the tumours that are only functional in the tumour tissue. And it's also possible to insert into the vector a promoter which is only activated in hypoxic or low oxygen levels, which allow you to only see CAR expression and CAR activation within the tumour microenvironment. The other approach which can help is also using allogeneic cells which do not have the associated cytokine release syndrome or neurotoxicity. They don't induce the same kind of immune response you see with alpha beta T cells, which is another approach that people are looking at. The other barriers are cost. So clearly taking the patient's own cells is a very expensive manufacturing technology. It's the majority of the cost of a CAR-T therapy in terms of cost of goods. It's costly and time-consuming, can be around 4 weeks from blood draw to re-administration of the engineered CAR-T cells into the patients. So an approach which gets around this is using allogeneic cells, which effectively are universal cells that can be used off the shelf. This can require the engineering of the T cells to inhibit MHC and TCR expression to avoid graft versus host disease. As mentioned above previously, it's possible to use naturally allogeneic cells such as NK cells and gamma delta T cells. This does reduce the cost. So, obviously one batch could treat many patients, whereas with autologous, you treat one patient with one batch. The product can be more homogeneous and there's less batch-to-batch variability, and it opens up the possibility of potentially redosing patients if required, and could accommodate larger doses which might be required for solid tumours. Another approach that people are looking at is actually developing in vivo CAR Ts. So in this type of product, there's no cell manufacturing process required. The vector has targeting moieties which allow you to directly transduce T cells in vivo via an intravenous injection. This does have a real potential to be a game changer in terms of radically reducing the cost of therapy, improving patient experience and expanding access to treat more patients and patients with early stage cancers. In terms of tackling solid tumours, which is probably one of the biggest issues, obviously limited efficacy to date in solid tumours. Tumours do express checkpoint inhibitors, which can modulate and exhaust T cells, reducing cytotoxicity and the ability to recruit other immune cells. And CAR T vectors can be engineered to express a number of different molecules. This could be inhibitors of checkpoint molecules to block the tumor-derived signals preventing T cell exhaustion, cytokines to ensure that the T cells remain activated within the tumor microenvironment, and also expressing chemokine receptors, which allow CAR Ts to traffic into the tumors stimulated by chemokines released from the tumor tissue. And the presence of T cell memory cells is actually a strong correlate of persistence and efficacy in CAR T clinical trials. And so there are ways you can enhance the number of TSCM cells in the cell culture. This can be using PI3 kinase inhibitors, which have been shown to increase the percentage of TSCM cells. And companies are also looking at other technologies which can potentially drive the TSCM numbers, be that through expression of factors within the vector or potential culture conditions which increase the numbers of these important cells. So in summary, CAR T's therapies have really shown impressive efficacy in certain hematological tumours. It's viewed by the field that we really do need to continue to invest in innovation to expand the use of this therapy into other patient types and other tumours. And the application of new technologies really does show great promise in expanding the use of CAR Ts in solid tumours and reducing cost, expanding access to patients with early stage diseases. So these CAR T therapies could become a mainstay of tumour treatment, of cancer treatment in the future. Thank you for your time, and I hope you enjoyed the presentation, and thank you.
Well, thanks for nice presentation. So your presentation shows the limitations of current CAR T therapies were very well analyzed. Thank you. 자, 다음은 이제 오늘 마지막 연자입니다.
I'd like to invite the last speaker, Dr. David Leung. He's the senior consultant with Merck Life Sciences, and he is in manufacturing and process development. He graduated from Australian National University, getting a PhD in organic chemistry, and is working with GSK for GMP manufacturing. He's going to talk about CAR T manufacturing process design and drivers for success. Are you ready, Dr. Leung? Are you ready, Dr. Leung?
Muted for a while. Let me just... Okay. All right. Thank you, everyone. And thank you, Bioplus, for the opportunity to speak to the audience. Today, my presentation is co-authored by my colleague, Antoine Aron, who is Head of Cell Therapy Commercial Strategy at Merck KGaA, and I am Senior Consultant for Novel Modalities Strategy at Merck KGaA. So the outline of today's presentation will be a focus on CAR-T manufacturing, a focus on the process. We look at what makes this process so complex, the complexity drivers, how to design an efficient CAR-T manufacturing process, and what tools might be available there are advanced manufacturing tools that can address some of these complexities of the CAR-T process. So this next slide shows that these advanced therapeutics are complicated. In the beginning, we had cellular therapy, and what this happened was that it provided antibodies in the form of antibody checkpoint inhibition to awaken a suppressed immune system. So antibodies would come in, disrupt certain interactions in our immune system, and then this would awaken our antibodies, our systems to fight the particular cancer. So the difference is that now for CAR-T therapies, what we're doing is we're educating naive T cells to be aware of how to recognize and target tumorous cells. So the way we do this is by cellular engineering. The way we engineer these CAR constructs is by using a viral vector or some other non-viral means. And these constructs themselves are actually quite complicated. They are transmembrane proteins. They have an intracellular domain, a transmembrane region, and then an antigen recognizing. So these very complex structures are difficult to manufacture, difficult to engineer. And this is one aspect. This is the molecular complexity of these CAR constructs. So what this means is that these urgent therapies have been rushed to the market. On the top here, you can see a conventional antibody development process. We have, about 6 to 7 years in terms of the whole development process. But because of the urgency of these therapies and the urgent unmet need and some of these accelerated regulatory pathways that are available for some of these indications, many CAR-T processes have a very compressed development timeline, as little as 4 and sometimes even less of years in the development pipeline. So what this means is that you have subpar processes that can be locked in because you need to get something to the patient very quickly. But these subpar processes then follow on and lead to high costs of goods because of various reasons I will describe later on. So how can the speed of process development with a high quality process development? So here is the second driver for the complexity in a CAR-T process. We have complex and labor-intensive workflows, and this results in high treatment costs. For example, we have separate plasmid, separate vector, and separate cell processes. You have your plasmid process, which is used in a transient transfection to make your lentivirus, or in some cases, your retrovirus. And then you have your cell process, which you obtain from a patient or a donor to get your patient or donor cells. These can be T cells or etc. Then you combine the viral vector with the T cells or the patient or donor-derived cells to get your CAR-T product. So you have this plasmid process, you have a vector process, you have a cell process, and then you have another cell process to get your CAR-T product. So it's a very complex of different processes in different facilities. You also have complex logistics for autologous therapies. For example, in an autologous therapy, you have your patient-derived cells that get processed and then given back to the patient within 2 to 4 weeks. It's slightly more straightforward generic product, whereas you have your healthy donors, and this can be banked a lot more control points in the allogeneic process. This can be banked and then given to the patient. So one of the when you look at the autologous process and you look at the cost of goods component analysis, this is some work taken from Spin and Stein's table, you can see that labor, this semicircle here, comprises the majority of the cost of goods for a CAR-T process. And out of that labor, manufacturing comprises the largest component. And then you also have your QC/QA, the me, as well as other components. So in the next slide, I will outline some aspects of how we can simplify these complicated processes. For example, we have robust platforms for plasmid and viral vectors. These are key raw materials for the CAR-T process. This can greatly secure this, simplify the complexity. For example, for the plasmid process, if we use a single-use operation, for example, with the cell lysis, the chromatography, or the ultrafiltration and sterile filtration aspects of these processes, this can greatly simplify the production of plasmid, which is a vital raw material for the viral vector process. On the other hand, we can also ensure that our upstream platforms to make the lentiviral vector and the viral vector types of viral vectors used for the CAR-T process have high titers and are suspension. So suspension means that you can scale up your upstream process for viral vector production. For example, a suspension adapted HEC293T cell line for high productivity, the use of PEI-based transfer for low cost of goods, chemically defined media in your upstream platform in order to assure process robustness, and also to ensure that whatever process, suspension process that you have is amenable to perform well at scale. So in terms, we can offer from Merck KGaA in our process solutions offering, we do offer single-use operations to purify the formulation and formulate plasmid. This is in our handbook for plasmid DNA downstream processes. We also have recently launched our Virus Express viral production platform with very high lentiviral titers. This is 5 E7 transduction units per milliliter. And this is for the reference GFP plasmid. If you would like to learn more about manufacturing platform development for viral vectors, please do check out our my webinar or webinar landing slide on the virus HEC293T lentiviral production platform. And this is a HEC293T clone that is optimized for transient lentivirus production in vector. This is already characterized and cGMP banked, so it's enough to start your upstream lentiviral process with a small vial of cells. Eventually, you will have to create your own cell bank, but most of the work for the cell banking is already done. It comes with the chemically defined media. This is a HEC293 chemically defined viral vector media. The whole process has been optimized for the clone as well as the media. And this process is also demonstrated at 50-liter bioreactor scale. And we're working on the subsequent and follow-up for this lentiviral vector production system. So when we've seen that in the manufacturing of cell therapy products that it's very complex, it's labor intensive, and also it's quite but when it comes down, if you want a robust process, you really need to know what is your product. And this stems from, first of all, characterizing your cell product, and this leads to quality by design. Quality by design then leads to robust processes. When we look at the wheel of what quality by design incorporates, it is this interconnection and this interweaving of both product knowledge as well as process understanding. One feeds on the other, and then when you unite all of these together, you have these continual improvement for your process. So therefore, defining your commercial end stage of your product quite early in your clinical development would support early process decisions towards more sustainable solutions at the end. First of all, we need to understand what is your product. So what is the quality of your product that's needed? What is the mechanism of action, and then this will lead to your critical quality attributes. And then we need to understand what is the manufacturing strategy. How many doses do you need at the peak penetration per month, per year, and which countries are you targeting and which geographies? Starting with the end in mind will lead to better process design. And finally, your process needs to be as simple as possible, yet as complex as necessary, because we are making complex products using a complex process. Of course, each project will have its own specificities and needs. For example, if it's autologous or genetic. And I would like to say that a robust manufacturing process really starts from the development of reliable product release testing methods. So, sorry. So therefore, aiming to build your product knowledge is key to understanding your product, its quality and mechanism of action. I'm convinced there is a lot to be gained by challenging your process workflow to identify steps that can be removed or combined in order to reduce the complexity of future operations in a GMP environment. So, as already said, a robust manufacturing process starts with product release testing methods to address 4 pillars around identity, quality, purity, safety, and potency of the products. When asking companies about their lessons learned from cell and gene therapy programs and what they would have done differently, many companies said that they would spend more time upfront in developing robust analytical methods. I would like to emphasize here our BioAlliance testing services for cell and gene therapies to help you build your product knowledge and biosafety testing strategy. Merck BioAlliance Testing Services is the leading provider of contract services in the biopharmaceutical industry, supporting every phase of test development and the manufacturing process, and offering a comprehensive and integrated package of services. We do develop specialized testing services for viral and gene therapy products like rapid sterility testing, replication competent lentivirus, integration site analysis by next-gen sequencing, for example. Our experts will be also delighted to help you navigate through the regulatory landscape. So here I've listed 3 pillars. We need to test for the identity and quality of CAR T cell therapies, the purity and safety of the cell therapy, as well as the integration site, which then leads to the potency of the particular CAR T therapy. So how can one manage a highly complex CAR T workflow? Whoops. Okay, here we go. So when designing a complex manufacturing template for cell therapy products, decisions will be impacted by the design of the facility and the existing capabilities of the CDMO partners. So key questions are, how do you build your template? Do you have a modular template or do you integrate a template? And which methods would you use for each processing step? Would you use an open step or can you close that step? Where to perform each processing step? Would you use a graded room, for example, A, B, or C, or would you use an isolator? And how do you secure the transfer of cellular materials under aseptic conditions from each step? Do bear in mind that for a cell processing workflow, you cannot do a sterile filtration to remove bioburden or microbial contamination because you would end up removing your cell product as well. So all these aspects are interlinked, and to simplify the process as much as you can, you gain more flexibility and opportunities to address these questions and also to design a more sustainable manufacturing process. So listed over here in the next slide are some tools that, and some criteria for tools that when you develop your manufacturing process. No doubt the future of cell therapy manufacturing will bring more templated processes, including solutions to close the process and to bring a more controlled environment through automation, new technology to gently process patient cells to ensure the highest quality therapeutic cells are then returned to the patients, technologies with ease of use, for example, intuitive setup and interface, and technologies that are scalable, capable of delivering consistent results, and to potentially buffer the variability of patient-derived materials. Ultimately, the goal is to make these therapies more affordable and accessible to as many patients as possible. So process innovation is required to fill all of these requirements. One example of this type of process innovation at Merck Life Sciences is the use of acoustophoresis to process the materials. Acoustophoresis is using ultrasonic waves to capture cells within an acoustic field by maintaining those cells in suspension. The first ECHO platform developed by Merck is used to concentrate and wash cellular materials in a fully closed and automated manner. ECHO acoustic cell process system has been tested across a wide range of cell types and applications. In a CAR-T process, this technology offers a lot of flexibility to achieve robust performance across a wide range of process parameters. Although the cell density can influence the process time, this technology can accommodate highly variable input materials, both in terms of volume as well as total of cells, as well as different methods for activating T cells to deliver consistent high viable cell recovery. This first animation here shows the two different types of activation used for T cells. We have Dynabeat and OKT3, and you can see that viable cell recovery as well as the process time required is roughly similar for both types of cell activation. This next animation here shows the difference between large volume processing, this is just under 1 liter, and then a low volume, high density processing, higher concentration at about half a liter. And in both cases, you can see that the viable cell recovery is quite high with a process time of just under 1 hour. The ECHO acoustic platform has been designed for autologous cell therapy processing in mind. It is part of the ECHO ecosystem, which also includes our cell selection device. So I'd like to close this presentation with 3 key learnings. First of all, the complexity of CAR therapies does make them challenging. By adequately characterizing the product, having reliable QC methods in place, and securing key raw materials, this enables the complexity of the CAR-T process to be managed. Lastly, disruptive platforms can streamline the industrialization of CAR-T, reduce process costs, and ultimately make these therapies more accessible to patients. So thank you very much. Please reach out to contact me on LinkedIn or via my email. And I'd like to close with some acknowledgements. Thank you very much to Antoine and Ron who helped put together this presentation with me. And thank you very much for your attention.
Well, thank you very much for your informative presentation. 자, 이렇게 해서 오늘 5
We have listened to the presentations. We had 2 Korean presentations, Epsilon and Terra Immune. And we heard from Oxford and Merck, including CAR-T. Cell therapies need CMC and manufacturing processes. I hope that today's presentations will be of great help to you in your future product development. With this, I'd like to end the second session, Breakthrough Technologies in Bio, and we will continue with a global clinical test in the 5th session, and Mr. Moon Han Lim, CEO of Cure Land Care Research, will be the moderator. Thank you very much.
Once again, thank you very much, moderator, Chairman Byung-gun Lee, and the 5 presenters.
Thank you for enlightening us. 바로 다음에는 글로벌 임상 세션 전에
Next, before the global clinical session, we will have a 10-minute or so break, and then we will have IQVIA sponsor session from 2:50. We will see you then.